For this video, we're going to look at a problem about what we call linkage analysis. So linkage analysis is similar to what we looked at when we look at Mendel and a dihybrid cross, in which we're looking at the inheritance of two traits. But when we're looking at a dihybrid cross like Mendel's peas, and we're looking at the inheritance of those two traits, those traits are inherited independently from each other because they are occurring on separate chromosomes, and so their segregation, or the how each of those genes are decided to put into which gamete, is going to be independent from each other. And so one trait has no influence on another trait in terms of the inheritance of those traits. And so we can, when doing those sorts of problems, we can work out probabilities for one trait, we can work out the probabilities of inheriting the other trait, and then because they're independent events, we can combine our probabilities and be able to come to some conclusion or make some predictions about the likelihood of having one set of traits versus another given any particular combination of parents. When we're looking at linkage analysis, we're looking at something a little bit different. Instead, what we're looking at is we're looking at a situation in which the chromosome, or the genes for the two traits that we're interested in, are occurring on the same chromosome. And so if we drew a chromosome, it's like having a gene, call it A, and having a gene, call it B, occur on the same piece of DNA. And so if, say, you have two different parents and they have different variations of gene A and gene B, they're homologous chromosomes, and we have, say, little a and little a, and then little b and little b, if in the production of gametes of this parent, for instance, is more likely that if you get the gene for the variant of big A or capital A, you're more likely to you're going to get the big B, because those are the capital B, those are linked together, they're on the same piece of DNA. When we think of what occurs during meiosis and where we have crossing over happen, where we have homologous recombination, where the maternal and the paternal chromosomes can exchange some information, that gives us the opportunity to rearrange or mix up these this sort of arrangement. And so we could end up having a crossing over event occur between these. And so in some instances of gamete formation, what we're going to get is we'll get 4 gametes where we'll have some gametes with the parental arrangements of alleles, where little a is linked to little b and capital A is linked to capital B. But in some of the gametes, we may see some recombination in which we get a different arrangement, and where we now have what we call recombinants, where we've rearranged the arrangement of alleles on the chromosomes. And so where most often, if you get the trait for the capital A, say it's a dominant trait, and the capital B is a dominant trait, if you inherit big A, capital A, you're more likely going to inherit capital B. And the same thing, lowercase a, or recessive trait, you're more likely to receive the recessive allele for B. But occasionally, you're going to get some recombination occur between these traits because they're occurring on the same chromosome. If they have if they're partially linked as opposed to completely linked. If they were completely linked, we'd get no recombination, no reshuffling. If they're on separate chromosomes, we'd call that independently assorting, and we would just be able to calculate the 50-50 probability of getting big A versus little a, the 50-50 probability of getting big B and little b, and then we could combine our probabilities. So when we're looking at a problem where we have two different traits and we don't get the Mendelian ratios, and so the Mendelian ratios for a dihybrid cross would be 9 to 3 to 3 to 1. And what that would mean is, when we read this problem, we see that the wild type eyes and wild type wings in these moths are the dominant trait, and that the recessive trait are these mutants where we have red eyes and white banded wings. And so if we were to cross in this case, where we had a homozygous for the dominant traits, for both dominant traits, and a homozygous for both recessive traits, cross to get our F1, and then that F1 is then crossed with itself, what we would expect to see is a 9 to 3 to 3 to 1 ratio. Okay, so we're not going to see that in a linkage analysis type of a problem because of the fact that these traits are linked together. And so kind of the key thing that's going to point to you that says this is a linkage problem as opposed to a dihybrid cross problem is a couple of different things. One is the fact that when we look at our ratios of offspring, what we're going to see is that we're going to see most often are the parental arrangements. In this case, we're looking at the F1 is crossed with a homozygous red eyes, homozygous white bands. Okay, and so the homozygous for the F1s, we're told, have normal eyes and normal wings. Wild type eyes, wild type wings is the same thing, and it's crossed with a moth with red eyes and white banded wings. That's red eyes with white banded wings. And so the thing that should jump out to you when to say, this is a linkage problem, is when you have those parental arrangements of traits are found as the most abundant ones in the offspring. And then when we mix them up a little bit, where we have the normal eyes going with the white banded wings, or we have the normal wings going with the red eyes that we have here in the middle, those are our recombinant offspring, which mean that we're mixing the traits up in a way that's different than what we see in the parents. Now, the other thing that would kind of hint to you that you're looking at a linkage problem was, one, we would probably say it straight in the problem, but if not, a linkage cross is done a little bit differently than Mendel did his dihybrid cross. For Mendel's dihybrid crosses, he would take homozygote dominants, homozygote recessives, cross to get our F1, and then he'd allow the F1s to self-fertilize, or he'd take two different F1s and cross those together to get our F2 generation. When we're doing a linkage analysis problem, it's a little bit different. We start the same way with the parental homozygote dominants with the homozygote recessives to cross to get all heterozygotes in our F1. But then we take that F1 and we cross it with a homozygote recessive in what we call a test cross. Okay, and so that is what we see here. Is this part of the prompt is laying out that we're taking homozygote normal moth, crossing it with a homozygote mutant moth, getting our F1s, the normal eyes and normal wings are in the F1. That tells us that normal eyes and normal wings are the dominant trait here. And then it says the F1 are crossed with moths that have red eyes and white banded wings. And so when we're looking at the abbreviations for these traits, we use RE plus for the wild type trait. And since it's dominant, RE plus with either another RE plus or an RE, the recessive, will get you wild type eyes. Okay. Whereas if you have two recessive REs without the plus, okay, this is going to give you red eyes. And if you have the white banded wild type allele, which is WB plus, the plus is indicating the wild type or the normal allele, and you have any other, whether it be WB plus or WB, you're going to get wild type wings. Okay, WT just stands for wild type or normal. Then if you get WB and WB, the recessive and the recessive, this is how you get the white banded wings. Okay. So here we're taking this F1 and we're crossing it with the homozygote recessive. So what we're getting here, what we're doing for a cross is we're taking RE plus and RE, WB plus, WB, and we're crossing it with a moth that's homozygous for the mutant alleles. Okay. And the first problem that we're asked is if these traits were independently assorting, what would the phenotypic proportions be? And so what we're looking at is what ratio of offspring would we expect if they were independently assorting? And so if they're independently assorting, what we can do is we would consider the inheritance of the RE gene, okay, or the eye color gene, and the inheritance of the wing trait independently, and we can work out the probability of each and then combine them to get the proportions of these phenotypic combinations. Okay, so ultimately at the end, what we want to know is how many of our offspring would we expect if these were independently assorting alleles, meaning they occur on separate chromosomes. How many offspring would we expect to be wild-type eyes with wild-type wings, red eyes with wild-type wings, wild-type eyes with white-banded wings, and red eyes with white-banded wings? Okay, so that's what we're looking for. And so to solve that problem, because we're stating that it's occurring as they're sorting independently, okay, or they're found on different chromosomes, is the same thing. Then what we can do is we can take each one independently, calculate the probabilities of getting each trait. What's the probability of getting wild-type eyes versus red eyes? What's the probability of getting wild-type wings versus the white-banded wings? And then we can combine our probabilities to find out what the likelihood or the proportions, relative proportions of what we would find for these combinations of traits. And so to do this, we can visually see it very easily by just doing a Punnett Square, where we take the gametes of one parent, the gametes of the other parent, and we have RE plus RE, RE plus RE, and then we have RE, 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 RE. And so this would be wild-type eyes here. And then these would be the red eyes. Since 2 out of the 4 options are wild-type eyes, we have a 1/2 or 50% chance of getting the wild-type eyes. Since 2 out of the 4 are the RE without the plus, meaning recessive, that gives us red eyes. We have a 1/2 probability of getting the red eyes. We can do the same thing when we look at the WB. We can say WB, WB, WB plus, WB. And we get WB plus, WB, WB plus, WB, 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 WB. And again, this would be the wild-type wings. And since we have half of the offspring are wild-type wings, we have a 1/2 probability or 50% chance of getting the wild-type wings. And since half of the offspring are recessive, or the white-banded wings, we have a 1/2 or 50% chance of getting those. So now what we need to do is actually answer the question, which is saying what are the expected proportions. So given these different phenotypic combinations that we see here above, if these were independently assorting, what ratio or proportions of the offspring would we expect in this cross if they were independently assorting? So now we can just combine our probabilities. And so we can just I'll just draw the results right up here, is if we were expecting for wild-type eyes, that's what we have right here, we have a 1/2 chance. For wild-type wings, we have a 1/2 chance, which gives us a 1/4 chance of getting wild-type eyes and wild-type wings. Same thing with the red eyes. For red eyes, okay, down here, we have a 1/2 chance. For the wild-type wings, we have 1/2. So we have 1/2 times 1/2 equals 1/4. Okay. Same thing with the wild-type eyes and the white-banded wings. Wild-type eyes, we have 1/2. White-banded wings, we have 1/2. So we have 1/2 times 1/2 equals 1/4. And then red eyes and white-banded wings, we have red eyes, white-banded wings. Again, 1/2 times 1/2 equals 1/4. Okay. So if these were independently assorting alleles, meaning they do not occur on the same chromosome, that they're found on separate chromosomes, then what the relative proportions from this cross that we would expect is 1/4 of them would be wild-type wings, wild-type eyes. 1/4 of them would be red eyes with wild-type wings. 1/4 of them would be wild-type eyes with white-banded wings. And 1/4 of them would be red eyes with white-banded wings. What that means is that we would expect our ratios to be equally proportional, in which that we'd get the same amount of offspring for each one. And what we see is we don't see that. Okay, and because we don't see that, that's what's going to tell us that this is an independently assorting trait. I mean, that this is a linked trait, that these are found on the same chromosome. And when we find two traits that are found on the same chromosome, what we're looking at instead is how frequently do we see recombination occur between those traits. And why this information is useful is because the frequency of that recombination, the primary thing that's going to be a function of is the distance those two genes are from each other. The further apart they are, the more often you're going to get recombination. The closer they are to each other, the less often you're going to get recombination. What this will ultimately allow us to do is if we look at several different traits and their relative rates of recombination, is we can start determining the relative order of traits going down a chromosome in terms of their linear positioning to each other. And we can also tell their relative distance from each other based on their rates of recombination. It's not a perfect science. It doesn't give us the actual physical distance in terms of letters of DNA, because other factors are going to play into the recombination rates. Chromosome structures, sequence, underlying sequence, things like that will also affect how frequently you see these recombination events occur. But in general, the primary thing that's going to affect the frequency of these recombinations is going to be the distance they are from each other. Okay. So now what that means is we can now use this information here rather than to work out probabilities and rather to try to make out predictions of the likelihood of getting one type trait versus another. What we can do now is we can now use this information to calculate a genetic distance. And to calculate the genetic distance, what we want to do is we want to take the number of recombinants that we have and divide in terms of progeny that we see here, the total number of recombinants that we have, and divide it by the total progeny. Okay, so the recombinants, remember what we started out with was a moth with wild type eyes and wild type wings. So that's a parental arrangement, crossed with a moth that had red eyes and white banded wings. So that's also the parental arrangement. So that is a parental arrangement. And that is a parental arrangement. Okay. Which means the two in between are what we call recombinant. All right. So now what we want to do is we want to add up all of the total number of offspring that we have, and we're going to divide that by the number of recombinants that we have. And so if we add up 222, 26, 24, and 228, we will get 500 total offspring. Okay, we're going to divide that by 26 plus 24 gives us 50 recombinants. And then we multiply that by 100%. Okay, and so what if you 50 divided by 500 gives us 10. We multiply by 0.1. We multiply by 100, that gives us 10% recombination. We can also say this as 10 centimorgans or 10 map units that we abbreviate as 10 MU. All of them would be equivalent. Okay, so that's how we can calculate recombination frequency using a test cross. Now, what can we do with this information? Well, that's what this other problem is designed to demonstrate. Okay, so now what we can do with that is we can do a series of crosses. So here we're looking at a series of crosses between different genes. Okay, so A, B, C, and D, they could be traits similar to what we see in the moth. So you got fruit flies, and one trait gives red eyes versus white eyes, one gives short wings versus long wings, one gives hairy legs versus not hairy legs, that sort of stuff. Okay, and so we can do a series of crosses where we're doing, say, red eyes and wing length, and red eyes, red and white eyes, and hairy legs, and work out the relative distance, the recombination frequency between each of the combinations of traits. Okay, you do lots of different crosses to get this done. And then you can create a chart like this that tells you all of the different recombination frequencies for those particular sets of traits. Now, one thing to kind of note here is that the ones that say 50, that is just as likely to be randomly assorting, okay? Because if we have randomly assorting alleles that are occurring on separate chromosomes, those traits you have a 50-50 chance of getting them. So randomly assorting or unlinked traits, we have a recombination frequency of 50%. So that will be the highest number you will ever see. Anything less than 50% will mean that those traits that have less than 50% are linked together. And so what you want to do is we're going to draw what's called a linkage map showing different linkage groups. Now, every gene should end up in its own linkage group. If a gene is not linked to anything else in the particular chart that you're looking at and the calculations that you've made, it still gets its linkage group because it's still a gene that's occurring on a chromosome somewhere. And so you need to account for that, and that's somewhere. Okay, so if we look through this chart, what we can see is we have A and B is 10%. Okay, so that means A is linked to B. Let's just kind of make some notes. A and B are together. Now A and C, 50%. So that means gene C is not linked to gene A. So we'll put it by itself. It should also not be linked to gene B. So if we look down here, gene B and C, 50%, yep, it's not linked to gene B either. Now A and D, A is linked to D. It's less than 50%, it's 14%. So D is also here. And then, which should also mean that D is not linked to C. And if we look at C and D, it's 50. So that means it's not linked. Okay. So now, what we want to do is draw what we call linkage maps or linkage groups. And so we could say C is in its own linkage group. Okay. It's on a piece of DNA somewhere, but it's not linked to any of these other traits A, B, or D. Now the question is the arrangement of these genes A, B, D, or is it A, D, B? Or is it D, A, B? Okay. What order do they go in? Okay, so that's what we're trying to answer. All right, so to answer that, we can draw another linkage group in which we put A, and then just pick any of them. You can, I've seen people suggest starting with the highest. Doesn't really matter. I think starting with the highest one is probably easiest. So if we start with A, and the A is the most distant from it is a D, we could put it on either side. We could put it over here, or we could put it over here. Doesn't matter. Okay, these are three-dimensional molecules. They can flip around, so it really doesn't matter. So we can put a D over here. And so that would mean the distance here is 14 centimorgans or 14 map units. So now what we want to do is now we have A and D, and now we need to put B in. So B, we could put B over here, or we could put B over here. Okay. If I put B over here and say that's 10 map units, well then that would say, we did that, then that would say that that's 24 from D to B, and when we look at from B to D, we get 4. Okay, so that can't be correct. Okay, so we know that B over here is not correct. And so if we put B in between, then A to B is 10, that we see right here. And then that would put D to B, 10 plus 4, it gives us 14, and that's what we get. So our linkage group looks like this, in which we have the gene order is DBA, with gene A being 14 centimorgans from gene D. Gene A is then 10 centimorgans from gene B, and B and D are 4 centimorgans apart. Okay, this chart could be as however many traits you're looking at. And you just do the same thing regardless of however many traits you have, and you just kind of keep working through it until you find, have accounted for all of the different traits and have all of the distances between them correct, and that's how you can draw a linkage map using recombination frequency. So hopefully you found this helpful. If you did, maybe give me a thumbs up to kind of let me know that this is actually working for you, and otherwise, have a great day.